Okay, so let's get started. So welcome everyone to today's Immune seminar. Again, this is absolutely my pleasure to welcome today's speaker, also my colleague, Pamela Schwartzberg she's the chief in cell signaling and immunity section in National Institute of Allergy and Infectious Diseases, NIAID. Pam received her bachelor degree from Princeton and her MD PhD training from Columbia College of Physician and Surgeon. So during Pam's PhD training, she worked on retroviral replication and early work using homologous recombination to introduce mutation into germline of mice, which absolutely groundbreak to help to open a new era in mouse genetics. I think a lot of us also benefit from her early time work. After an internship at Boston Children's Hospital in Boston, Penn actually did a fellowship at the National Cancer Institute studying tyrosine kinase signaling pathways as a special fellow of Leukemia and then Lymphoma Society, which I think we're going to hear she's going to talk about today. Penn actually started her own laboratory at National Human Genome Research Institute at the end of 1997 and was promoted to senior investigator with tenure in 2003. Her lab's work has centered on the use of genetics, biomedical, cellular and genomic studies to understand T-cell signaling with a focus on pathways affected by primary immunodeficiency. The work in Penn's lab focused on T lymphocyte signaling with an emphasis on pathways that are affected by primary immunodeficiency and then how they regulate normal immune cell development and function. They examine functions of signaling molecules in T cells, mainly focus on tyrosine phosphorylation initiated pathway. Currently, Penn's lab work have several different directions, which include they study a mouse model of activated PI3 kinase delta syndrome to provide insight into how PI3 kinase regulates expression of TCF1 and the maintenance of long-term CD8 T cell responses. And then also they try to target CRISPR using CRISPR screenings in primary mouse T lymphocytes to discover how signaling pathways regulate T cell function, adhesion, and the migration, and then T follicular help T cells generation. Eventually, their work also complemented by the study of human immunology, focusing on response to immunization and to infections with SARS-CoV-2, which Penn significantly contributed to this scientific society about COVID research. So Penn currently is a adjunct faculty member at the University of Pennsylvania and then the Georgia Washington University School of Biomedical Sciences, and it has received several NIH awards for mentoring, who actually really is spectacular mentor. I think a lot of trainees benefit from Penn's guidance in science, and also I believe a lot of things in personal life. Penn has served on numerous mentoring, reviewing, and then editorial boards, including she's a recipient of a Serial Scholars Award and then the American Association of Immunologists BD Pharmaging Biosciences Award for Early Career Scientists, and then has been elected to American Society for Clinical Investigation and the Association of American Physicians. But I think among all the achievements Penn got all these years, I think one thing I want to particularly focus or emphasize is Penn's really a great mentor. I think, as I said, her lab actually nurtured a lot of great trainees who's been really successful and activated in both academia and then industry. So it's absolutely our pleasure to welcome Penn on our platform. We're very much looking forward to your talk.
Thank you, Tron. That was for such a nice introduction, really. I feel honored. And it's a pleasure to be here today. So let me just switch. And so what I'd like to tell you about today is some of our work looking at a disease-activated PI3 kinase syndrome and how that gives us windows of insight into the immune system. And so my lab has had a long interest in T-cell signaling, T-cell receptor signaling, and particularly how T-cells receive and integrate signals from the environment, including signals from the T-cell receptor, from co-stimulatory receptors, and from cytokines to lead to the development of downstream effector function. And that can be cytolytic function or development of distinct types of effector type T cells. And one of the amazing things about these signal transduction pathways and the molecules involved in them, and this is actually a very gross simplification of some of these signaling pathways. But what's amazing about these pathways is how many of these molecules are mutated in human disease. And that can include primary immunodeficiencies where T cells don't function properly, as well as cancer and overgrowth syndromes. And so despite many of these molecules having being involved in signaling downstream from the T cell receptor, they actually can give rise to a wide range of different types of diseases. And the questions we've been interested in are how do signaling pathways affect distinct types of T cell function and responses to infection? And what can primary immunodeficiencies and genetic models of them teach us about immune system function? And so I'm going to focus my talk a little bit more on T follicular helper cells and humoral immunity, or the development of successful antibody-mediated immunity and how T cells contribute. So the disease I'm going to really talk about today is a disease called activated PI3 kinase delta syndrome that results from activating mutations seen in a heterozygous form. And we first got interested in this disease when a talented clinician in the NIH Clinical Center contacted us about a group of patients who had come into the clinic there who showed signs of immunodeficiency, and they were actually referred in for chronic EBV and CMV infection. We now know that this group of patients, actually only about half or less of them have chronic EBV, but they all have recurrent respiratory and gut infections, and they also show signs of immune dysregulation, including blood lymphopenia, yet lymphadenopathy and splenomegaly, and altered lymphocyte populations, autoimmune manifestations, and altered responses to immunization. Now, when Gulbu performed whole exome sequencing on these patients, what she realized is that many of them harbored heterozygous mutations affecting P110 delta, a hematopoietic specific catalytic subunit of PI3 kinase. And we've become interested in this disease over the years, and particularly one of the first things we focused on was the fact that these patients had abnormal antibody levels, particularly higher serum IgM, variable levels of IgG, but impaired responses to certain kinds of vaccination. Now, we've been interested for a while, actually, stemming from work we did early on with Shane Crotty and with Hai Chi, looking at T follicular helper cells. Now, these are the CD4 subset that help orchestrate humoral responses through interactions with B cells that help provide signals for antibody isotype switching or class switching, as well as formation of the germinal center. And that's the site where you're going to have somatic hypermutation of your immunoglobulin genes and affinity maturation. So when T cells get activated by a foreign antigen and in the context of MHC by an antigen presenting cell, they'll upregulate this chemokine receptor CXCR5. They'll migrate to the B cell zone of the lymph node, where they'll have cognate interactions with B cells, long-lived interactions that lead to their migration into the B cell follicle and providing signals both from the B cells to form T follicular helper cells and from the TFH cells to form this germinal center structure. And this is really where you're going to get your generation of high affinity, long-lived memory B cells and long-lived plasma cells. Now, these TFH cells are marked by expression of this chemokine receptor, CXCR5, as well as PD-1 and ICOS, and they're regulated by this master transcription factor, BCL6. So how does PI3 kinase fit into this? Forward. So PI3 kinases are lipid kinases that regulate many key functions in lymphocytes. The class 1 PI3 kinases are heterodimeric proteins that are activated downstream of generally tyrosine phosphorylation-based signaling. And what they do is they add a phosphate to this ubiquitous membrane lipid PI45-PIP2 to generate PI345-PIP3 or PIP3. And that is going to serve as a molecule that will recruit proteins containing plextrin homology domains or other domains that bind PIP3. And these molecules get recruited to the membrane. The classic molecule is AKT, which gets phosphorylated by another pH domain containing kinase, PDK1, and then gets activated. That in turn will lead to phosphorylation of a wide range of downstream targets, leading to regulation or activation of mTORC1, regulation of GSK3, and regulation of transcription factors such as FOXO1, BOC1, that when phosphorylated are actually excluded from the nucleus. So these transcription factors are turned off. And together, this conspires to regulate metabolism, growth, survival, differentiation, migration, and adhesion. And when we start to think about TFH cells, what we realized is that many of the receptors that regulate the interactions between TFH cells and B cells, and they're important for the formation of germinal centers, including the T cell receptor, but also ICOS, are very good activators of PI3 kinase. And so Silvia Prete, a talented fellow in my lab, who's now a group leader at AstraZeneca, decided to look in the cells from these patients. And you can look in the peripheral blood from these patients and look at the expression of CXCR5 on activated T cells. And that's a surrogate marker for circulating TFH-like cells. And what we could see is actually in these patients, we saw increased TFH cells, even though they had poor antibody responses. So to try to understand what was going on, we actually made a mouse model that expressed the most commonly, a mutation equivalent to the most common mutation found in the patients. And this is a E1020K mutant. And in fact, these were a great model of the disease. The mice were lymphopenic in the blood, yet they had lymphadenopathy and splenomegaly. They had more activated T cells, fewer naive T cells. And they develop these mucosal lymphoid follicles in the lung and the gut. And I forgot to say that early on, that one of the distinctive features of this disease is actually that these patients have these mucosal nodules, lymphocytic nodules in the gut and the lung. And we see the same thing in the mice. And all of these phenotypes worsen as they age. We see increased T follicular helper cells, just like in the patients. And now, because we can look in the lymphoid organs, because you can't, that's the germinal center is a structure that's formed primarily in the secondary lymphoid organs, you can see that they had increased germinal centers. And they also had increased plasma cells, the antibody producing cells, and they saw increased IgM and IgG. That's a little bit different than in the patients. So why do they have these increased TFH cells? And we think there's both cell intrinsic and extrinsic mechanisms. And so for cell intrinsic, one of the receptors that's really great at activating PI3 kinase delta is ICOS. And ICOS is critical for the formation of T follicular helper cells. And you can see that here, if we take an OT2 T cell receptor transgenic model, we transfer them into wild type mice, we immunize the mice with NP-OVA, and then we look to see, are they forming TFH cells marked by CXCR5 and PD-1. And you can see the wild type cells form TFH cells quite well, but if we block ICOS ligand interactions, we prevent that formation of TFH cells. Now, PI3 kinase, as I said, is going to activate AKT. AKT can phosphorylate the FOXO transcription factors, and FOXO1 is a very potent repressor of BCL6. And so if we do that same experiment using activated PI3 kinase T cells in the transfer, you can see, number one, we get more TFH cells, so cell intrinsic phenotype, but they're barely blocked by the addition of anti-ICOS ligand into body. So it's ICOS independent development of these. If we look at T cells from these mice and stimulate them with anti-CD3 and CD28 antibodies, we see increased phosphorylation of AKT and increased phosphorylation of that FOXO transcription factor, which will lead to its nuclear exclusion and turning off. Furthermore, if we look at the effects of ICOS, what we can see is if we take activated T cells and stimulate them through the T cell receptor in blue, or T cell receptor plus ICOS, and we look at FOXO1 by flow, we can see that ICOS stimulation potentiates the phosphorylation of FOXO1, consistent with its role in activating PI3 kinase. But in the PI3 kinase delta mutants, we see not constitutively, but we see a high induction, even independent of ICOS. And so we can bypass the requirement for ICOS to phosphorylate FOXO1, which would be required to de-repress BCL6. And furthermore, we can take a mutant of FOXO1 that cannot be phosphorylated by AKT. And what we can see is that lowers the percentage of TFH cells, both in wild type and in the mutant. But are these TFH cells functional? And so to try to address this, we looked at where they were and looked at the germinal center structure. And so in wild type cells, in wild type mice, germinal centers have a very discrete function consisting of a light zone and a dark zone. And the light zone is where the TFH cells are located. You can see that here marked with B cells, with PD1. And you can see this increased TFH cells in this dark zone. And this is where the B cells and T cells will be getting their interactions, giving signals for the B cells to migrate to the dark zone where they undergo proliferation. And that's why it's dark, there's more cells there. However, in the activated PI3 kinase mice, what we see is that, in fact, there is this poor demarcation of the light zone and the dark zone, and there's infiltration of the TFH cells throughout the dark zone area. So there's increased germinal centers, but they're disorganized with altered light zone and dark zone ratios, increased TFH cells in the dark zone. Some of these phenotypes are actually B cell intrinsic, including a decrease in B cell death that likely contributes to the increased germinal centers, and also a defect in class switching to IgG1. And as a result, if we immunize these mice, even though they have higher levels of immunoglobulin, they get poor antigen-specific responses post-immunization. So what are all the serum immunoglobulins if they're not getting specific responses? Well, in fact, many of them are autoantibodies. And so if we look at it, use an autoantigen array and put on the serum from wild type or the PI3 kinase, activated PI3 kinase mice, and compare it to a known lupus model strain, what you can see here is the lupus-prone mice have very high reactivity to a wide range of autoantigens, but the activated PI3 kinase mice also have a significantly elevated auto reactivity to multiple antigens seen on this array. Now, we also, if we look in the gut, we can see that they actually, if we take bacteria from their autologous fecal pellets, we can see that we also see increased reactivity against autologous fecal bacteria. And in fact, many of these antibodies also cross-react with self-antigens. Now, this is not because I took out that data. It's not because they have altered gut microbiota as far as we can tell at a gross level. But in fact, what we see is that both the B cells and the T cells seem hyper responsive to microbiome extracts from their gut. And in fact, they're hyper responsive, even the B cells, to TLR stimulation. So if we see the same thing with stimulating these cells with LPS, you can see that the PI3 kinase mice will show increased expression of activation markers. They'll proliferate more than the wild type, and that can be blocked by adding a PI3 kinase delta inhibitor. And so that raised the question, if some of this is hyperreactivity to gut bacteria, what happens if we treat these mice with antibiotics? And so we did that starting at weaning for 5 to 6 weeks. And what we saw is we could absolutely prevent the development of autoantibodies, suggesting that in fact commensal stimulation may be essential to trigger some of these autoreactive phenotypes. And so we think that this activated PI3 kinase delta is just too much of a good thing. And that in fact, the PI3 kinase delta is a critical gatekeeper, and the proper regulation of that actually permits generation of appropriate humoral responses in the face of commensal stimulation. And so I'm not going to be talking about CD8 responses today, but in fact, in multiple situations, what we see is that we get less of a specific response and more of a non-specific response because of the continual activation of these cells. But what I want to talk about today is perhaps how else may activated PI3 kinase delta affect humoral immunity. And so one of the things that T cells need to do in order to develop TFH cells and to provide cognate signals to B cells is they need to be able to have long-lived interactions with B cells. And you can see that here, looking at the interaction of a T cell and a B cell target here, or an antigen presenting B cell. And that interaction is critical for the development of TFH cells and germinal centers, particularly work we've done with Hai Chi, looking at another immunodeficiency involving a molecule known as SAP and the SLAM family members. But in fact, what seems to be critical for mediating that interaction between T and B cells is interactions involving the cell surface adhesion receptors known as integrins, in particular for T cells, LFA1. And so integrins, as I said, are cell surface adhesion receptors. And when they're not adhesive, they're actually in naive cells are in this low affinity state. But once the T cell gets triggered through the T cell receptor or through chemokines, it introduces a signal transduction cascade that leads to a change in the conformation of the integrin with a change in the extracellular domain that now allows these integrins to become more adhesive, in this case, bind better to its ligand ICAM. And data suggested that PI3 kinase, data from Klaus Okenhag's lab, had suggested that PI3 kinase was part of this signaling pathway. So how can we look at this? Well, we can actually use a flow-based flow cytometry-based ICAM binding assay, where we can stimulate cells through the T cell receptor and then look for LFA1 binding to ICAM1 that's fluorescently labeled. And we actually use a multimerized ICAM because then we can look both at changes in conformation and also changes in aggregation or avidity. And so you can see right here, if we do this assay in wild type, you stimulate through the T cell receptor and you can see an increase in ICAM binding, a rapid increase that then slowly decays over time. But if we do the same experiment in cells from our hyperactive PI3 kinase delta mice, what you can see is this very high binding of ICAM that really stays high. Conversely, if we look in a kinase dead mutant of PA110 delta, we can see that they would see suppressed ICAM binding. Now we can reproduce the same thing using CRISPR-mediated immunogenesis of these molecules. And so again, here's the wild type control. Each experiment looks a little bit different. If we knock out PI3 kinase delta, we reduce the binding. We can see that this binding is really mediated by LFA1. If we look at these few cells that bind ICAM, they're still expressing LFA1. Conversely, if we knock out this lipid phosphatase, which actually counteracts the activity of PI3 kinase by removing the D3 phosphate off of PIP3, so this is P10. If we knock out P10, we can see, again, this elevated binding. So how does PI3 kinase affect inside-out signaling, as this is called when we increase the adhesion of LFA1? Well, the signaling pathways downstream of TCR that lead to inside-out signaling are complex. They involve the SLP76 complex, but also direct interactions from ZAP70 that lead to the recruitment and activation of this RAP1 GTPase, which when active binds the RAP ligand, and that can bind to the intracellular tails along with tailin of the of LFA1 and lead to the change in conformation. And RAP1 is also involved in downstream signaling, or what we call outside-in signaling. How does PI3 kinase affect these pathways is not clear, but it's interesting that many of these molecules involved in this pathway actually have plexurin homology domains. It's not clear that they all bind PI3, but they potentially have maybe involved in PI3 kinase's effects. So to try to look to see what PI3 kinase effectors and what molecules are important for inside-out signaling, Chris Johansson, a talented graduate student in the lab, who was joint with Klaus Okenhaupt's lab as part of our OxCam Welcome collaborative graduate program, decided to try to tackle this by doing a targeted CRISPR screen. And so what Chris did was he bioinformatically curated a list of all the pH domain containing proteins, only a fraction of which are expected to or predicted to bind PI3. But that's very poorly defined, so he included all of them. And he also included any other PI3 protein that had been shown to have PI3 binding activity in the literature. And he used this list of genes to create a library of CRISPR guides in a retroviral vector. And then he used that to transduce cells, primary cells, grow them out, and then a week later, perform our ICAM binding assay. And what he could then do is sort out the cells that didn't bind ICAM from the cells that did bind ICAM and determine the guide RNA enrichment in each population by next-gen sequencing. And when he did that, actually had beautiful results. It was a very clean screen. And what you can see is actually the top hits were our control, which targeted the alpha chain of LFA1. But we also saw very strong effects of knocking out Kindlin3 and RIAM. Those were two of those proteins that are known to be part of the integrin signaling cascade. We also saw effects of knocking out tailin of PI3 kinase delta, as we might expect. And then we saw a number of really interesting things, including proteins that, a number of mitochondrial proteins, which is interesting to think about in terms of energy required for adhesion. Conversely, at the other end of the spectrum, if we looked at genes that if we knock them out, they increased ICAM binding. Our top hit was P10, our control. But then the next hit was this molecule RAS-A3. Now, RAS-A3, or IAP4 binding protein, or GAP1, was described previously and has been particularly studied in platelets, a cell type that really is dependent on integrin activation, as a RAS-RAP1 gap, or GTPase activating protein. So RAP1 is a GTPase protein. When it's in its GTP bound form, it's active. When it's in its GDP bound form, it's inactive. And RAS-A3 helps catalyze that GTPase activating. It's a gap. And so it's going to turn off RAP1. Okay. And so if we were able to get RAS-A3 conditional knockout mice from Wolfgang Bergmeyer's lab. And if we look at those cells and we stimulate T cells from those mice with anti-CD3, what we can see using a probe that looks at active RAP1 GTP, what we can see is, so this probe only binds active RAP1. What we can see is in response to TCR stimulation, you can see this increased in RAP1 GTP. But in the RAS-A3 knockout, in many experiments, we can see that RAP1 is constitutively active and its activation is elevated. As a result, if we look at those cells, we can see compared to wild type in black, both in naive and in activated cells, you can see that the RAS-A3 have, in red, have elevated ICAM binding using our ICAM binding assay. You can see that difference is particularly profound in the naive cells. You can also notice that naive cells bind less ICAM than activated CD4 cells. And part of that is LFA1 is upregulated. But part of it is also that in naive cells, activation of integrins is somewhat suppressed. So why are the effects so different? Well, in fact, if we re-express RAS-A3, and here we're overexpressing RAS-A3, what we see is that we can actually suppress ICAM binding, and we actually suppress it down to levels that look similar to the naive cells. And if we look at expression of RAS-A3, it becomes perhaps clear why this may be. So RAS-A3 is highly expressed in naive cells, and then it's rapidly downregulated at both the message and the protein level. And so within hours, you see downregulation of the message. This is actually looking at 24 hours. You can see how markedly it's suppressed. It's also suppressed at the protein level. And this is looking at Doreen Cantrell's proteomics database IMPRESS. Now, the same type of activation, so in fact, it suggests that that part of the reason why naive cells may be less, or their LFA1 may be less easily activated, is because you have this high level of RAS-A3. Now, this increased ICAM binding is not just seen in response to TCR signaling, it's also seen in response to chemokines. Again, taking naive cells and stimulating them with different chemokines, we can see this increased ICAM binding. If we look at these T cells, they're actually hyperproliferative, particularly at low CD3 doses. Yet if we look at TCR signaling itself, if we look at phosphorylation of ZAP70, it looks equivalent. If we look at phosphorylation of ERK, if anything, it might look a little bit down, but yet these cells show this increased activation of RAP1. And that suggests that in T cells, RAS-A3 is primarily affecting RAP1. So it's primarily acting as a gap for RAP1, not necessarily for RAS, which would affect ERK. So how does PI3 kinase affect RAS-A3? So if you think about it, RAS-A3 is going to turn off RAP1, yet PI3 kinase turns on RAP1. And so you can see, if we look at activated RAP in stimulated wild-type T cells, or stimulated cells that have a PI3 kinase inhibitor, you can see that inhibition of PI3 kinase prevents full activation of RAP1. But if we do the same experiment in the RAS-A3 deficient cells, they're not affected. And so that tells you two things. One, that RAS-A3 seems to be the main PI3 kinase effector required for activation of RAP1. And because RAS-A3 turns off RAP1, it suggests that PI3 kinase must inhibit RAS-A3 activity. Now, if we look at a mutant of RAS-A3 that cannot bind PIP3, so this is a plextrin homology domain mutant, you can see that after TCR stimulation, RAS-A3 is mainly out of the plasma membrane. You look with this pH domain mutant, you can see that that localization shifts and is primarily cytosolic. And then if we look at the function of that mutant, we see something quite interesting. So if we take our RAS-A3 deficient cells here in red, you can see their ICAM binding. We re-express wild-type RAS-A3 in this experiment, it's suppressed. But if we re-express the pH domain mutant, it's even more suppressed. Whereas if we use a gap minus mutant, it looks no different. It looks like we haven't transduced anything. So the pH domain mutant that can't bind PIP3 actually works better. And so it really suggests that RAS-A3 is a novel PI3 kinase effector that's inhibited by PI3 kinase activity and by its pH domain. And that's really a novel mechanism for how a protein with a pH domain acts. Most proteins with pH domains get recruited to the membrane where they get activated. This molecule seems to be inactivated by PI3 kinase. So what do these mice look like? Well, so we have them crossed to a CD4 Cre, so they're going to be deleted for relatively late in development, and thymic development looks relatively normal, except we noted that it did look like there were slightly more single positive cells, so the most mature thymocytes. And if we look within that single positive thymocyte population, looking at a marker CD24, it's downregulated in your most mature population, we can see that actually is those most mature cells are increased. If we look in the periphery, we actually see the converse. So it looks like maybe these cells are being held up in the thymus. So they're extremely lymphopenic in the blood. They're also lymphopenic in the spleen and the lymph node. Interestingly, no effect on the mesenteric lymph node or the Peyer's patches in terms of T cell numbers. And that's intriguing because gut homing actually involves a different integrin, alpha-4 beta-7, that binds MADCAM1. And there's data both from Tim Springer's lab a long time ago and more recently from KInase's lab arguing that RAP1 activation may not be required for the initial activation of alpha-4 beta-7. So there's slightly different requirements there. Perhaps that's why there's no difference there. What about the function of these cells? Well, as we said, TFH cells provide contact-dependent help for B cells for germinal formation. And we had previously seen in work we had done with Hai Chi that if you disrupt long-lived TB cell interactions, such as in the absence of SAP, it prevents full differentiation of TFH cells and germinal center B cells. So here we have more adhesion. So what's going to happen? So we immunized mice with NP-OVA and used either alum or Adavax as adjuvants. And what we saw was actually these mice had decreased germinal center formation. So decreased germinal center B cells. They also had decreased class switching, which is TFH cells are important for. So you can see here in wild type, you have more IgG1 positive cells, fewer IgM positive cells that are recognizing NP. And the immunogen, whereas in the RAS-A3, we have more IgM positive cells, so they're not class switching as well. So you have slight reductions actually in the high affinity IgM, but actually very dramatic defects in NP-specific IgG. If we look at TFH cells, we can see they're also reduced in number, but interestingly, they're actually increased in the percentage of CD4 cells. And we can actually see that these TFH cells here, color in orange, we're looking at LFA1 levels. And so you can see how high the levels are in TFH cells, both in wild type and the mutant. So a higher percentage of the CD4 cells, but lower numbers. So if we look at the germinal centers, they have germinal centers. I think they look a little funky, but everything we tried to measure looked relatively normal. And if we look at the percentage of those germinal centers, that B cells that can bind NP, the antigen, it's absolutely normal, but the numbers are down. If we look to see the relationship between TFH cells and the germinal center B cells, for both the mutant and the wild type, it pretty much follows a linear relationship. There's just fewer cells there. So what happens if the cells are normal? And so to address that, we looked in the Peyer's patches and the mesenteric lymph nodes where we do get T cells there. And then the germinal center percentages are normal. So it suggests that these cells can differentiate into functional TFH cells if they're in the lymph node. The same is true in vitro. In fact, both in vitro and in vivo, as a percentage, they actually differentiate better. But if we transfer CD4 cells, we still get low numbers getting into the lymph node, have lower numbers of germinal centers. And that really suggests the problem is getting into the lymph node. I will say if we transfer CD4 cells, we do rescue the class switching defect, and that may be due to, it may be effects of the TFR cells. I'd be happy to talk about that in questions. So what does lymph node entry look like? So to address that, what we did was we did T cell transfers of both wild type and congenitally marked mutant cells and looked either immediately after transfer or at longer time points. And what we could see is immediately after transfer, we actually saw that this is looking at a ratio of the knockouts to wild type. So we actually see more cells in the blood, fewer cells in the lymphoid organs. After 24 hours, this starts to normalize, but even after 48 hours, we always see lower percentages of cells in the peripheral lymph nodes. If we look to see where these cells are, interestingly, if we're looking in the high endothelial venules, and so these are where cells are going to enter the lymph nodes, what happens is T lymphocytes they'll sort of see signals from certain adhesion molecules, and will start to roll along the inside of these venules. And then in response to signals from chemokines, they'll actually start the process. They'll activate their integrins and start the process of extravasation. And so what we see is we actually see plenty of cells in the high endothelial venules. There's just fewer cells in the lymph node parenchyma. And so we can actually quantitate that over time. You can see that there's this buildup of cells in the high endothelial venule. So to try to look at this better, we look by, and when I say we, this was Chung Park and John Curl's lab. We looked at this process of entry into lymph nodes using multiphoton live imaging. And so what we can see is that the RASA3 and the wild type cells, let's look at a RASA3 knockout, they start this process where they start to migrate through the endothelial cell layer, but then they look like they get trapped. And so once again, they start this process, but then they get trapped on top of the basement membrane, and they don't continue to migrate into the lymph node. So you get this accumulation in the walls of the high endothelial venue. Now, some cells can migrate normally. You can see here's a cell entering, both a mutant and a wild type. See how they're entering into the lymph node parenchyma, but many of these cells are trapped. So what's really going on here? And so when we looked at these images, we realized that they looked very similar to what had been described by Mark Bazhanoff and colleagues as high endothelial venules. High endothelial venule pockets. And these he described as waiting areas for lymphocyte entry that help determine lymph node size. And so if you prevent egress of lymph node cells from the lymph node using S1P inhibitors, so cells can't get out, what you see is that lymphoid cells T cells start to start the process of transendothelial migration, but they actually get stuck here in these waiting areas above the endothelial barrier. And so what it looks like is that our cells can start the process. They actually start it quite well, but then they can't turn off integrins, and they can't really complete this process. And so that ability to both turn on integrins and turn off integrins is really critical to complete transendothelial migration and regulate lymphoid cellularity. Well, we've also shown that RASA3 affects lymph node egress, and that may be why we see a comparative loss of cells from the blood over time. So what we've really seen is that by trying to understand how PI3 kinase regulates cell adhesion, what we've done is uncover a novel regulator of T cell integrin adhesion and trafficking. This is an interesting molecule that's inhibited by PI3 binding and PI3 kinase activity, a really novel mechanism of regulation. Its expression is very dynamically regulated. That's high in naive cells, but very rapidly turned off. But it's not only highly expressed in naive cells, it's also highly re-expressed in memory T cells, which is interesting to think about in terms of migration. And it work when we had just uncovered this work from Yi-Sun Wan's lab at UNC had shown that it's actually highly expressed in pathogenic Th17 cells, and that in the absence of this molecule, you don't develop EAE in mouse models of multiple sclerosis. So obviously this has important implications for T cell trafficking and function, particularly for tissue-specific phenotypes in the CNS and gut. And its phenotypes really highlight the critical requirement for T cells to regulate adhesion tightly, not just by activating it, but also being able to turn it off. Now, the RAS-A molecules have not just been highlighted by our work and the work on pathogenic Th17 cells, but also in the last year, work from Alex Marson's lab has uncovered another RAS-A family member, RAS-A2, in CRISPR screens looking for molecules that boost antigen sensitivity and long-term function. And what he did was he did multiple orthogonal CRISPR screens looking for knockouts that will allow cells to proliferate and get activated in response to, despite having the presence of multiple inhibitory molecules, such as cyclosporin, adenosine, and TGF-beta. And one of the only two hits he came up with was RAS-A2, a molecule that's very similar to RAS-A3, but is more specific for as a RAS gap. So it turns off RAS and the RAS MAP kinase pathways. And so together it suggests that these RAS-A molecule proteins may actually be novel gatekeepers of T cell activation that help keep naive T cells off. And when they're downregulated, again, they also saw rapid downregulation of RAS-A2. It allows T cells, so once you activate a T cell, you downregulate these RAS-A proteins, and it allows T cells to become fully activated. So a very interesting series of molecules, again, that have come out of some of our studies of PI3 kinase. So I want to take the last few minutes just to switch gears, and I may go over just a teeny bit, and talk about some of our work in humans, and particularly trying to understand these same types of adaptive immune response is in lymphoid tissues, but in response to SARS-CoV-2 infection. And so many papers, hundreds of thousands now, have examined adaptive immune responses to SARS-CoV-2 infection, and most of the studies of convalescence patients have really focused on peripheral blood, with few exceptions. And so early on in the pandemic, work from Shiv Pillai's lab, although this has now been repeated by multiple groups, is that if you look in post-mortem samples from people who've died of COVID and look at the thoracic lymph nodes, what you can see is there's actually a disruption of germinal centers and a striking reduction of TFH cells. However, more recent, and furthermore, critically ill patients were also shown not to seem to develop a germinal center response as well, but really to develop what's called an extrafollicular response, a kind of development of B cells that's been previously described in autoimmune and chronic infection settings. However, more recently, work from Ali Elabedi and Donna Farber's lab have actually shown evidence of long-lived plasma cells, which are derived from the germinal centers, work from Ali Elabedi, and actually germinal centers in the lung lymph nodes from organ donors that had happened to have evidence of having SARS-CoV-2 infection. So that suggested that germinal centers really can be formed, and that, I think, is the consensus now. But at the beginning of these studies, it really wasn't clear, and it's really difficult to access most of these tissues, because if you want to look at germinal centers, the site where you're really going to get your critical long-term humoral immunity, you need to look at lymphoid organs. However, tonsils and adenoids are mucosal lymphoid tissues and secondary lymphoid organs of the upper respiratory tract. They're poised near the site of infection and viral replication and contain populations not present in peripheral blood. And that includes germinal center B cells, TFH cells, and tissue-resident memory cells. Unlike many of these other tissues, they're accessible tissues. Tonsillectomy and adenoidectomy are the second most common ambulatory surgery in the US. And so Kalpin M. Anthiran, assistant clinical investigator associated with my group, and Sandy Zhu, a postdoctoral fellow in my group, decided to try to look at, could we look at responses to SARS-CoV-2 in the tonsils and adenoids and evaluate localized immunity to the virus? And second, to understand how SARS-CoV-2 affects the immune system in convalescence. And this was in collaboration with Pam Mudd at Children's National Medical Center. And I should say that Kalpana Manthiran has really been studying tonsil biology and had a strong collaborations with these groups. Now, so what they did was they collected tonsils and adenoids and blood from 110 participants in the fall of 2020. So this is really pretty early during the pandemic. So September 2020 to January 2021, we collected most of these, the first 100. We had to do it blindly because all of the patients had to be PCR negative at the time of surgery, or mostly blindly. And so what they did was they collected the samples and performed serology and high dimensional flow cytometry. And by doing that, they found that 24 of these patients actually had evidence of prior COVID infection. Only 11 of these by questionnaire had known that they had been infected, but the other 13 we found by evidence by serology and flow. So really, again, many of these patients did not know. And the ones that did know, that they were from a range of 25 days, and then we collected the last 10 patients in the spring of 2021. And so we had one very long-term, a couple of long-term people there. I should say that these people in gray didn't know that they were infected, but these three down here actually came for surgery, turned out to have SARS-CoV-2 infection, active infection, were sent back home. They were PCR positive, and they were sent back home for a few weeks and then came back in and were negative and underwent surgery. And so we use these samples for serology and high dimensional flow cytometry using the Cytek Aurora. We also looked in a number of samples by CytSeq imaging and performed functional assays. So what did we see? So all of these patients had neutralizing titers against early strains against COVID, lower percentages to Delta, and even lower percentages to Omicron, which was not a circulating strain at that time. If we looked at those titers to the ancestral strain, we could see that they decreased over time. But then we could use probes against the S1 a protein, the spike protein. And what we could see is that we could see in the peripheral blood, but also in the adenoid and tonsils, we could see spike-specific B cells. And if we looked at those cells, those memory cells, over time, they did not decrease. And furthermore, we could see that particularly the percentage of spike positive B cells in the adenoid actually correlated with serum titers to many of these strains, suggesting really the adenoid was where we were seeing the critical action. If we looked at what these cells were that were spike positive, you can see that the bulk of them are memory in green, but you can also see in the adenoid and tonsils, we could detect germinal center B cells. And if we look by flow cytometry, if we look by histochemistry, what we could see, and this was done by Andrea Racki and Ron Germain's group, we could see that those adenoids and tonsils had beautiful germinal centers. So we did not see any disruption of the germinal center structure. And furthermore, we could see that if we looked at the percentage of germinal center B cells that were spike positive, RBD positive, it was higher in the adenoid, again, suggesting the adenoid, which is in the nasopharynx as opposed to the oropharynx, really was perhaps where more of the action was in response. So then we took cells from a few patients and looked by CytSeq, where we can look simultaneously by single cell RNA-seq at the gene expression and look at proteins on the cell surface. And so when we do that, what we could see is we could separate, if we perform unsupervised clustering, we could see distinct clusters of B cells. And what we could see is that the bulk of the spike-specific cells fell into two clusters. This cluster 2 here, which had a gene expression signature of memory B cells. And that's what we saw in the tissue. We also saw another CD27 positive IgD minus memory B cell population that by cell surface markers we would describe as memory B cells. This was cluster 9, but if you look by gene expression, it was actually quite a bit different than the cluster 2 that we saw. So memory cells in the periphery and memory cells in the organs in the tissue may look somewhat different. We could look within this cluster too and look at the spike-specific cells versus the spike negative cells. And within that, the spike positive cells had higher expression of CXCR3, a chemokine receptor that's induced by interferon gamma, as well as HOPX, another interferon gamma induced gene, suggesting that exposure to that viral antigen. And remember, all of these cells are convalescent, in patients that are convalescent. We could look at the spike-specific cells, and we saw that they showed high percentages of class switching, more IgG than IgA, even in the tissue, and they had high percentages of somatic hypermutation. If we looked at their diversity, we could see that the spike-specific cells were less diverse than the spike-negative, suggesting they were undergoing clonal expansion. And in fact, if we looked specifically at the BCR sequences, which we could do in the single-cell level, what we could see is that there were clones or lineages that overlap between the adenoid and the tonsil within a given patient. And we could actually then look at that and look at somatic hypermutation and class switching, and we could see that there were clones that you could find both in the adenoid and the tonsil. And in some cases, you could see here a clone that was in the germinal center, and that was that cluster 4, as well as in memory cells. And so we could detect cells both in the germinal center. I should point that out here. There were a small percentage of cells in this cluster that were spike positive, and that was a germinal center cluster. So we can see actually migration of cells between these two organs. So then, what about how does SARS-CoV-2 infection affect the overall immune landscape in the tissue in the blood? And so to do that, we looked at our high dimensional flow cytometry and looked by unsupervised clustering and said, what kinds of cells do we see, and do they change post COVID-19? So once you've had that, do you see long-lasting changes? And so what we could see first off is that there was a reduction. If you were positive, you saw reduced naive B cells. But what we did see was we saw increased percentages of cells that were germinal center B cells, both IgM and IgG positive germinal center B cells in people who had COVID, particularly in the adenoid. If we looked in the T cells populations, we could see increased percentages of certain TFH cell populations. So again, looking in the adenoid, we could see a reduction in this cluster 9, which was a naive population, whereas we saw increased percentages in cluster 3, and cluster 3 was a PD-1 CXCR5 positive population that also expressed CXCR3. We also, and consistent with that, we could actually see these cells in the increased percentages of the cells in the germinal center. Now, the other thing that this cell population also expressed was CD57. And CD57 is a marker that goes along with, it's often seen in chronic infections. It's associated with senescent type T cells, but it's also a marker of TFH cells within the tonsil. And now we see in the adenoid, and you see increased percentages of these CD57 TFH-like cells within the germinal center. Now, consistent with these cells, with the phenotypes we saw in the B cells, we also saw increased percentages of CXCR3 positive cells. And consistent with that, if we just stimulated T cells from these patients, what we could see is we saw increased percentages of cells that produced interferon gamma in the COVID convalescent patients. We also saw increased percentages of cells that expressed IL-21 and IL-2, which are consistent with TFH cells. Some of these populations also expressed IL-10. So what did we see actually post-infection? What we saw is persistent changes in the tonsils and adenoids after COVID-19. So we saw increased germinal center B cells. We saw increased percentages of these CD57, PD1 germinal center TFH cells, as well as CXCR3 positive TFH-like populations. We also saw increased CD57 and CXCR3 positive populations of resident memory CD8 cells. And all of these, we saw more changes in the tissue than in the peripheral blood. So we saw these enriched populations involved are located in the germinal center or associated with tissue interferon gamma responses. So what about antigen-specific T cells? Well, to do that, what we did was we took cells from the blood, we stimulated with different peptides from different COVID proteins, and you could see that we could see some COVID-specific cells, T cells. And if we concatenated all those cells and looked at their properties and compared that to total CD4 cells, we could see that these COVID-specific cells were enriched for CXCR5 PD1 TFH cells, and those TFH cells were enriched for CXCR3 positive populations compared to the total T cells. So we see this enrichment for specific cells. Now, in the organs, we had too much background. We could not do this kind of activation-induced marker assay. So what we did instead was we went back to our site-seq data, and we looked specifically in activated T cells that we had sorted out. And what we could see is in cells that were from COVID convalescent patients versus the naive controls, what we see is that we had a higher percentage of expanded T cell clones. And so if we looked at the phenotypes of those T cell clones, again, in the peripheral blood, they were CD57 positive TRM cells. These are CD8 cells. And in the tissue, they were CD57. These were CD57 PBMCs, and these were CD57 TRMs in the tissue. If we looked at those sequences and compared them to SARS-CoV-2 specific databases, we could see that many of these clones overlapped with SARS-CoV-2 specific TCR sequences, both directed against spike and against ORF. And furthermore, we could also see overlap of clones between tonsil and the adenoid. So again, that same kind of theme that we're seeing SARS-CoV-2 expanding specific populations, and these seem to be SARS-CoV-2 specific cells, and we can see them migrating among tissue. So why some of these cells have expanded for months? So why is that? Well, in fact, if we look with Dan Chertow's lab looking at viral RNA, we can actually see that persists for months in the tissues of certain convalescent patients. Suggesting whether this viral persistent, whether that indicates antigen persistence, we don't know. We could not detect that. So what did we learn from this? Well, one, we had very high rates of seropositivity. Early in the pandemic, in fall 2020, we already had 20% positive of children in the Washington, D.C. area. And we saw evidence of robust SARS-CoV-2 specific memory and germinal center B cells in the oropharyngeal tissue that was class switched B cells with somatic hypermutation and overlapping clones in the tonsils and adenoids, showing that you can get great germinal center responses. We saw persistent changes in the tonsils and adenoids, and particularly the adenoid changes correlated with neutralizing titers. What that says about reactivity of adenoids versus tonsils is not clear, but certainly suggests that these are interesting tissues for looking at responses. What the implications are for protective immunity and pathology in children, I think, are really interesting questions. But then the final question I just want to talk about, I know I'm at the end of my time, was how does this look after vaccination versus infection? And so to try to do that and really ask the question, does vaccination generate mucosal immunity, particularly in the oropharynx? And so what we did, and how does this compare to natural infection? And that's particularly important because we are learning more. We're seeing that natural infection may confer longer lasting immunity. So in 2022, we collected another 21 patients that were vaccinated. We haven't had high uptake of vaccines in kids. Of these, when we tested by serology, we found that 10 of them were vaccinated alone. And so if we look in the blood, we can see SARS-CoV-2 specific B cells in both the infected and the vaccinated. Vaccinated show higher levels in the blood, but amazingly, we can also see cells, SARS-CoV-2 specific cells in the vaccinated in the adenoids and the tonsils. And these are mostly memory B cells, but they don't all look the same. In fact, in the blood and in the tissue, what we can see is the infected have more IgA positive cells. We can also look but using high dimensional flow cytometry. And in fact, if we look, this is just in the blood. If we look at the different subpopulations of B cells, we can pull out. What we can see is that these subpopulations in the tissues and the blood, in both the blood and the tissues, look different between the infected and the vaccinated. So even in the blood, in particular, one change I want to highlight is if it looks like, again, the vaccinated have more CXCR3 positive cells. So I've just circled two clusters, these IgA positive clusters. If you look in the infected, they have more of this cluster 9, which is CXCR3 positive, whereas the vaccinated IgA positive cells are mainly in this cluster 13. That cluster is a CD20 low, atypical or activated type memory cluster. And we see the same type of thing in the tissues. I want to point out that again, in the tissues, we can even see a few cells that are in the germinal center clusters. So post-infection versus post-vaccination, we could see cells in the tissue, but they look different, both in the blood and the tissue. With post-infection, more IgA, more CXCR3 positive, post-vaccination, more IgE and more atypical markers. How do these memory B cells differ? Do they have different functional characteristics we're trying to look at? What are the implications for protective immunity and evaluating vaccination strategies? And how do T cells compare in these organs are important questions. As I mentioned, this is really the work of two talented people, Sandy Zhu and Kalpana Manthiram in my lab group. I also talked about the work of former member Sylvia Prete and Christopher Johansson, and the current work on PI3 kinase is being done by Jennifer Cannons and Dom Golick. So sorry for going over and sorry for some of the glitches there. Thank you again for the invitation.